Hello everyone! Today, we're going to learn how to extract DNA from green peas. For this experiment, you will need 1/2 cup of split peas, 1/8 teaspoon of table salt, 1 cup of cold water, 2 tablespoons of dishwashing liquid, a pinch of meat tenderizer, 70% cold isopropyl alcohol, a blender, a strainer, and a small glass. In the blender, add 1/2 cup of split peas, 1/8 teaspoon of table salt, and 1 cup of cold water. Blend on high for 15 seconds. The blender separates the pea cells from each other, so now you have a really thin pea cell soup. After that, pour the thin pea cell soup through a strainer and into another container. Add 2 tablespoons of liquid detergent and swirl to mix. Let the mixture sit for 5 to 10 minutes. Gently pour the mixture into a small glass until it is about 1/3 full. For the enzyme, we will add a pinch of meat tenderizer. If you do not have a meat tenderizer, you can also use pineapple juice or contact lens cleansing solution. Tilt the glass at an angle and slowly pour the cold isopropyl alcohol down the side of the glass so that it forms a layer on top of the pea mixture. Pour until you have about the same amount of alcohol in the glass as the pea mixture. Using a wooden stick, slowly stir the mixture. On the alcohol layer, you will see a bunch of white stuff clumped together. This is now the extracted pea DNA. In this experiment, we have followed the 3 fundamental steps in DNA extraction: cell lysis, DNA extraction, and DNA precipitation. The first step is cell lysis. The DNA of the pea cells is enclosed in cellular membranes and enclosed again inside the tough cell wall. Cell lysis involves the breakdown of the cell wall and the cell membrane to release the cellular contents and ultimately the DNA. Blending the peas helps separate the pea cells from each other and break down the cell wall. The dishwashing liquid contains compounds that removes fats and proteins. Because the membranes surrounding the cells are made up of fats and proteins, the dishwashing liquid will pull apart the membranes, releasing the DNA. Once the DNA has been released, we will now separate it from other cellular components and proteins associated with it. Straining the pea soup separates most of the cell debris and the mashed pieces of peas. Because DNA is still tightly wound, it is so small it slips through with the liquid and into the glass. DNA is associated with proteins such as histones. To remove this, we use a protease enzyme in meat tenderizer to degrade the proteins. After DNA has been isolated, we will now precipitate them so that they would form visible clumps. The salt that we added neutralizes the negative charge on the negatively charged phosphate in DNA and thus enables the DNA strands to stick together. It also causes proteins and carbohydrates to precipitate. However, the water molecules that surround the DNA create a protective shell that makes it difficult for the salt ions and DNA molecules to interact with each other. The addition of isopropyl alcohol helps remove the hydration shell of water molecules around the DNA, thus allowing the DNA to precipitate. The alcohol used must be cold because if the alcohol is too warm, it may cause the DNA to denature or break down. DNA is insoluble to alcohol, thus adding it helps DNA clump together and form a visible white precipitate.